In this video we will discuss about genes, that is what is inside behind the genes and how actually the is transformed into the functional protein. Gene is basically a specific sequence of nucleotide that is present in the DNA, and this is the DNA shown here, and it has different sequence of nucleotide that is correspond to a particular gene. And within the sequence of nucleotide we need to understand that some part is recognized as exon and some part is recognized as intron, and, and we will understand what this exon and intron means, and uh, out of these two which one is important. So here the DNA sequence is being shown in a straight line. So P, the initial point, is denoted as promoter. Before it, it's a nucleosome, and after that, some part of the DNA is exon, and then intron, and then again exons. And we need to understand that exons are the coding sequence, and introns are the non-coding sequence. So in the final product, in the process of forming the final protein, we need to get rid of introns. So let us first understand what is shown in the left figure. Here the co-transcriptional splicing is shown, that is during the transcription of this DNA to RNA, the intron removal takes place during this transcription step. That's why it's called co-transcriptional splicing, and this is done by the polymerase RNAPI2. This RNAPI2 will bind to the intron, and it will form a splicing complex by attracting this splicing sequence, and it will cleave off the introns at this 5' splice site and this 3' splice site, and in this way the intron is removed from the DNA sequence during transcription step. And at the same time, this RNAP2 also helps in initiation near the promoter, and all simultaneous and parallelly it also helps in elongation of the exons, so that the transcription will continue uh, throughout the exons. And one more important function this RNAP2 does is it helps in the termination, and it helps in the termination, that is it cleaves off the polyadenylated mRNA, and we know that this polyadenylated is essential for the stability of the mRNA. And figure in the right side shows another function of the intron. In this case, what happens is that the cleaving of intron helps in the interaction of this promoter and termination sequence, and this helps in acquiring the final protein to get their three-dimensional structure, as it can shown in the panel B of the right side figure, that the promoters and the termination sites interact with each other to form a loop that helps the proteins to acquire a final three-dimensional functional structure. And in this case what happens is that the splicing of the, as it is shown written here, is the promoter 5 splice site interaction early during the transcriptional site, that is splicing of this site happens first, instead of splicing of both the sites happen at the same time. This helps in the formation of this loop, and at the same time it also helps uh, in bringing together the promoter and the termination, uh, so that they can help each other in forming a final functional native structure. And here it is shown the interaction between the promoter and termination in the final figure, that promoter site, 5 promoter 5 prime splice site interaction with terminator 3 prime splice site, and this all this are all this complex formation is supported by RNAPI2 polymerase. So this RNAPI2 plays an essential role in the transcription of DNA to RNA, and then allowing the final proteins to acquire their native structure. I hope you have understood some functioning of this, uh, how the gene is been finally how the gene is converted into its final product or the final protein, and what are exons and introns, and which of them are important at this moment. At this moment, the complete function of the intron is not yet been known. However, several theories has proposed different function of the introns, but a general consensus has yet to be made on the functioning of intron. That's all for this video. Thank you for your time.